Respiration, Unit 3, Lesson 3. Miss Ray here. We're going to be learning about this process that all living things do so that they can harness energy for their cells. So you should have your packets ready to go and be ready to write. So there's a couple questions that we are going to answer about this process. So first off, who? All living things do this process, or at least start this process, to be able to harness, again, energy. What is it? It's a process that releases the trapped chemical energy that has been stored in food in the form of glucose, where it then is going to break it down in the form of with the presence of oxygen and turning it into ATP. Remember, in photosynthesis, plants or autotrophs were able to harness sunlight and turn it into first chemical energy and then later on a carbon molecule energy form in the form of glucose that was used for storage. This process takes that carbon molecule glucose that's stored in food that is gotten from when organisms eat plants, and then it reverses it and turns it into chemical energy in the form of ATP that cells can use. Because cells can use ATP, they cannot use glucose. Now, when does this occur? Well, cells need to have this occurring all the time because, well, cells need energy all the time. And this is going to mainly occur in the mitochondria. That's the main organelle. But there is a step that does occur in the cytoplasm. Now, here's our generic equation. We have our chemical equation and also our compound equation. So we take glucose and oxygen, glucose from the food that we eat. That's the C6H12O6. And we combine it with oxygen. Those bonds are broken. And then we are producing carbon dioxide, which we breathe out. We also breathe out a little bit of water. And we'll talk about that a little bit later, how that works, to then produce what the main product of cellular respiration is, which is ATP, that energy. Now notice that the reactants and products of cellular respiration and photosynthesis are opposite. So what is needed to create what's needed by plants, for example, is what's produced by cellular respiration, and what's needed by cellular respiration is produced by plants in again, photosynthesis and cellular respiration. So they are reverse processes. Now let's learn a little bit more about the mitochondria. So this is a membrane-bound organelle that helps regulate energy. We call it the powerhouse of the cell. And again, cells have these so that they're able to metabolize the glucose, break it down into energy. Now, there are lots of cells that have this organelle. And because the mitochondria is the main organelle of cellular respiration, all cells are able to do some process of this process. Now, you have an image in your notes that I'd like for you to take a moment to identify the different parts. I want you to star the inner membrane and the matrix because those are the two main locations that you will need to know within the mitochondria. Now, cellular respiration is broken up into 3 steps, 3 processes. Technically, at the collegiate level, you'll find out that it's a little bit more complicated than that, and there's actually a 4th one, but for high school, we're just going to simplify it to 3. The first step is glycolysis, and all living things do this process. We're going to find out later that they don't always finish with the other 2, but all living things can do glycolysis. And this is going to first start outside of the mitochondria, in the cytoplasm. And it is an anaerobic process. We'll talk more about what anaerobic means, but it really just means that oxygen is not used, it's not there, it's not present when this process is occurring. And what's happening is it's taking one glucose and it's breaking it into something called pyruvic acid. And when they're breaking these bonds, because remember there's lots of bonds in glucose to be broken, they're releasing energy. And they're releasing energy into the forms of smaller energy, ATP and NADH. NADH is not the same as NADPH. So keep that in, sort of remember that. When we get to our unit test, that sometimes trips people up. NADH is specific to cellular respiration, NADPH is specific to photosynthesis. So again, our inputs, our reactants, is one molecule of glucose. And weirdly enough, we have to use ATP to actually jumpstart this process. The output is we're going to produce 2 molecules of pyruvic acid. We're also going to produce 2 NADHs and 4 ATPs. Now, when we talk about the net ATP, that means at the end of the process, what we walk away with profit. Remember, this is an energy, how much energy can our cells actually use? Well, remember that there were 2 ATPs that we had to use to jumpstart it. So when we take the 2 that we need to jumpstart it, even though we produce 4, we only really get 2 ATP as our product that we can actually use that profit margin. And again, this is sort of a visual image of taking that glucose, breaking the bonds so that we have 2 smaller carbon molecules called pyruvic acid that then are going to enter the mitochondria. Now, once glycolysis is finished, we're now going to send that pyruvic acid into the mitochondria. And weirdly enough, it's going to go into the center of the mitochondria, immediately into the matrix first. And this is going to then take our pyruvic acid and it's going to break it up even more and break it specifically into CO2. And while that breaking of bonds is happening, NADH is being created, FADH2s are being created, and ATPs are being created. Now, because at this point there is oxygen in the mitochondria, because it's going to be needed later, this is considered an aerobic reaction, which means it does happen in the presence of oxygen, even though we don't need it. Again, inputs to pyruvic acid. There's a small change where we take that pyruvic acid and we make it a little bit smaller. So it can actually fit into the mitochondria. We turn it into something called acetyl-CoA. Main, though, takeaway is we need to know that pyruvic acid is what we start with, 2 molecules of it, and then we end up producing our CO2. This is where the carbon dioxide that we exhale, this is where this is created. We also create all these other byproducts we create ATP, NADH, and FADH2s, and we create 6 NADHs and 2 FADH2s. FADH2 is a new molecule. We haven't heard of that one yet. This is another energy-carrying, electron-carrying molecule that is going to become needed, but it's carrying little bits and pieces of energy. Again, our end result at this point, we've made 2 more ATPs that cells can use. And now we're on to our last one. So we can sort of see here where the Krebs cycle happens within the matrix of the center. And what's going to end up happening is that all of the NADHs produced by glycolysis and also by the Krebs cycle, and all the FADH2s produced by the Krebs cycle are going to be sent to the mitochondrial inner membrane. So this is where the ETC, or the electron transport chain, occurs. This is the third process. Again, it's the inner membrane spaces where this is occurring. They're going to take all those high electron carrying molecules, all the NADHs, all the FADH2s, and they're going to strip them of the energy. They're also going to utilize um, they're going to utilize those hydrogens and break those bonds for those hydrogens. They're going to be combined to make water through utilizing the oxygen that we breathe in to then charge up even more ATPs. This is the main ATP producing process in cellular respiration. So again, high electron energy molecules, NADHs and FADH2s are sent into this. The electrons, they're electron carrying, energy carrying, hydrogen ion carrying, and they're going to be stripped of those ions. The bonds are going to be broken, and it's going to be used to charge up more ATP. So, of course, we need NADHs and FADH2s, 8 of those and 2 of those respectively. We also need oxygen. This is where the point of breathing in oxygen is here. We need something to be able to carry these NADs and FADs out once they've been broken. So they're going to be combined. The hydrogens are going to be broken off, combined with oxygen to make water. Because we know when we take hydrogen and we take oxygen and put it together, it makes water. And specifically, it makes water vapor. I want you to do a little bit of an experiment right now. I want you to take your hand, put it right in front of your face, and I want you to breathe really heavily on it. You're going to feel some moisture on your hand. That is the water vapor that is produced in this process. Now, this net ATP has a range, and at the smallest, it produces typically 26, and at the greatest, it produces 32. And it's because sometimes it's more efficient, sometimes it's less efficient. And so we have this sort of range that we need to know. The also important thing is because of this range, there are a lot of things online that do not have the same ATP net numbers. So you're going to want to make sure that you're using our note versions and not anybody else's versions. So this is just a little image where we take our FADHs, the hydrogens are broken off from them. The hydrogen ions are sent across the membrane. Water is produced. And then they take those hydrogen ions, and those hydrogen ions are then used to charge up ADPs into ATPs. And then that's what, again, we want to make sure that we can utilize for our cells. So aerobic cellular respiration, pretty much one glucose goes in, and we end up getting a range of 32 to 36 ATP and water to come out. That makes sense because if we add up 2 from glycolysis, 2 from the Krebs cycle, and anywhere from the 28 to 32, we would get our total net ATP for the whole process would be 32 to 36. We talk a lot about the role of glucose prior to this point in this lecture video because we're needing to start glucose with the process, and we've been breaking down those bonds and all that. But oxygen is also a really important molecule that's needed for this process. And we utilize 2 words, aerobic and anaerobic, meaning the presence of or having or needing oxygen versus not needing oxygen to help separate our 3 processes. Because oxygen goes into the mitochondria and stays there, even though it's only actually used by ETC, it's still there while Krebs cycle is doing its thing. So both the Krebs cycle and the ETC are considered aerobic reactions. Versus anaerobic reactions, glycolysis does not need oxygen, nor is there oxygen just sitting in the cytoplasm, so it is considered an anaerobic process. Now, the reason we want to generate this is that there are organisms that do not require oxygen or don't live in places that have enough oxygen to be able to complete cellular respiration. And this is where there are these processes that can harness a little bit, make a little bit more ATP, but not go into the Krebs cycle and the ETC. So we're going to talk about anaerobic respiration starting now. So anaerobic respiration is a process that does not require oxygen, and it can be performed when oxygen is not present. It's the very first step is going to still be glycolysis. Glycolysis is very needed. All organisms do glycolysis, and it's going to occur still in the cytoplasm. If an organism does not have enough oxygen, then, if an organism has enough oxygen and has a mitochondria, they're going to continue the process into the Krebs cycle and ETC. But if they don't, they won't. And instead, they'll do two alternative forms of fermentation. So we call an organism that we call the process when glycolysis is just used, and then we turn around and either make ethanol or lactate. We call this process fermentation, and typically the product is going to be producing a lot of NADHs. And what's really happening is we're taking the pyruvic acid that was made by glycolysis, and it's being broken down so that then glycolysis can start again. Now, there's two types of fermentation. You have lactic acid fermentation. This is usually done by fungus and bacteria, but actually, weirdly enough, humans can also do this process too. We only do it though when we need a burst of energy, not a lot, like we're talking like less than 90 seconds worth of energy. Typically, it's when we're doing a high amount of cardio. It's the tail end of the game, or we're at the very tail end of a sprint, something like that. That's when we sort of see that process happen. It is not enough for our cells to actually be able to run on. So again, cell respiration, the full 3 is what our cells really need us to do. Now, alcoholic fermentation is the other type, and this is usually done by yeast and also some bacteria. And we harness that process a lot. We do a lot of food creation utilizing bacteria and fungus and yeast to create a lot of interesting things. For example, when we are creating bread that rises, we're using a lot of times yeast to do that. Yogurt uses a lot of bacteria. We make a lot of alcoholic beverages using bacteria, and, like wine and beer and other kind of stuff. Kimchi, any kind of pickled items of some sort, uses alcoholic fermentation in that process to create those foods. Now, we have ended our notes. There are 2 parts of practice. There is the image one, where I want you to use the word bank to fill in the parts of cell respiration that you can sort of figure out. And then you're going to take the total numbers that we've been collecting in our notes and see if you can't figure out how ATP is made from the NADHs and FADH2s. So just for walk yourself through all of this. All right, this is the end of your notes. It can be tricky, so make sure that you are taking the time to study and ask questions when you get to class.